So our next speaker will be Anasua Mukhopadhyay, and she's actually from the Adolf Merkle Institute, which is in the Swiss Freiburg, or Fribourg, anyway you want. So when I met her, she was a postdoc with Michael Meyer, and she now moved on to be a junior group leader. Congrats, by the way.
Thank you so much.
Yeah, and she will not be reporting about like optical readout, but they rather have tailor-made nanopores, quite large nanopores, which they then can use on these orbit devices as basically detection devices, for example, for disease-related agents. Yeah. So please, Anasua.
Yeah. Thank you, Conrad, for the nice introduction, and also thanks to Jan because for his great talk, and also my job is a little bit easier now because he has already introduced protein nanopores, so that's good. Okay, so I'm Anasua, and today I'll be talking about single molecule measurements, what to do with protein nanopores, for mostly looking into biomarkers of neurodegenerative diseases. This is us in Freiburg in Switzerland, and part of our group focuses on individual protein biomarker detection for various diseases, for example, Alzheimer's, Parkinson's, Huntington's. Today, I'll mostly focus on Alzheimer's. A little bit of introduction before I go into details. So if you compare the brain cells or the brain of a person with Alzheimer's and with a person who has like all normal healthy brain, you'll see quite significant changes. First of all, the size of the brain has shrunken a little bit in the right side, and also you'll see this massive holes appear in the brain. Why so? Because the cells in the brains have died, and unlike all our human cells in our body, which regenerate with time, we have to take care of our brain cells because it doesn't. And after some time, if you have all these diseases, it creates all these holes, and which finally creates all your loss of memory and all different sorts of dementia. Now, there are two main pathological hallmarks of Alzheimer's, and these is amyloid plaques and tau tangles, which looks under a normal microscope like this, fibrillar structures. But it's very important to mention here that we all have tau protein in our brains, which helps in keeping the microtubule structure intact and help us function. But only in the cases of Alzheimer's, what happens is the protein gets hyperphosphorylated, and in this way, it gets detached from the microtubule, and then we lose many of our healthy functions. And there are all advanced techniques to look at them, this in detail of these tau tangles. So if you take an electron microscope and look at these tangles in a zoomed-in view, you will see all the tangles are made of this side of fibrillar structures, which in the community we call them amyloid fibrils or amyloid plaques sometimes. And if you zoom them, and if you try to understand why and how exactly the mechanism or why exactly this forms, so it starts with soluble monomers or monomer proteins, and then it tries to clump up different like monomers and form this sort of structures, small aggregates of protein, which we call protein oligomers, and then it forms all this kind of fibrils, which results in tangles. Now, in last, if I was giving this talk like let's say 30 years before, people are mostly focused on the fibrils, and they thought that these are the most toxic species. It's still a very controversial topic, which are toxic in all these neurodegenerative diseases. But now in recent 10 years, many of the scientists in the community believe that these earlier oligomers or the small protein clumps are responsible, or maybe majorly responsible for the diseases, because since they're small, they can travel inside your cells, and they can catalyze larger or make very large assemblies, which are toxic. So today's talk is, and then it's also important to mention here, there are different type of amyloid forming proteins, for example, alpha-syn, and all these different proteins, but tau is one of them, which is responsible in Alzheimer's. And today's talk is mostly on tau. Now, before we go into why we are doing this and why suddenly the nanopores are important for tau, is if the major, like there are different techniques exist in currently to characterize the biomarkers for neurodegenerative diseases. For example, ELISA and also FRET is very successful in characterizing these biomarkers. But what the lack is for the measurements, they always have to chemically change the modifications, or for nanopores, what they do is they generally chop the protein in smaller parts and then they characterize. So what our intention is, we want to analyze all these biomarkers, all these protein aggregates without any labeling, without any modifications. And it's very important that we want to measure this from a heterogeneous samples, because this will ultimately help us in analyzing all the real samples. And then you want to analyze what exactly happens, how the population changes. Now, what we do on a day-to-day basis in our lab, I'll not go into details because Jan has already mentioned, but we generally paint bilayers using these amazing orbit setups, which made our life really easy, I would say. We'll never go back to the traditional setups. But yes, we paint bilayers, and then we form protein channels or protein nanopores. And then we, whenever we see a single step insertion or a current jump, we can measure the diameter of the pore, and we call it a single pore. And we always take one single pores, not multiple pores, for all our analysis. And once we have a pore, we add our analyte, or here the proteins from top channel, which we call a cis channel, and then we see them passing through the nanopores, and we study the current blockages. And you can see that if there is a large molecule present, it will block more current. And from the amplitude of the current, we can analyze the particle diameter, or we call it volume or particle sizes. So it exactly looks like this. If a large particle passes through the nanopore, it blocks large currents. And if you have an anisotropic molecule, which is not spherical, it blocks the currents. The current modulation is different. And by studying the current modulation, we can study the aspect ratio or length to diameter ratio of the protein. Now, when we started this project, let's say last 2-3 years back, we look for nanopores which are large in diameter. Why so? Because since our focus is mostly on the neurodegenerative diseases, it forms large molecular structures. So all the existing pores till later last year was mostly 5.5 nanometer to 0.7 nanometer. They were quite very amazing pores, but exactly not behaving for our purposes. So we were looking for large diameter pores, and beginning of the year, we have published the first large diameter nanopore, which we call polyC9 nanopore, with internal diameter 10 nanometers, which can successfully characterize a lot of single proteins from 0.5 kilodalton to, let's say, 100 kilodaltons. This is all good, but when we took up the challenge of analyzing tau, the tau is 42 kilodalton in size, and if you form large aggregates, it's huge, massive. So we wanted to have like a large diameter pore, and then we came across this beautiful cryo-EM structure from Katharina van Viesgrove, where they have shown in bacterial cells the assembly of pneumolysin, and it creates huge 25 nanometer diameter pores, which are very effective for killing cells. So it's not so viable, but or let's say biological sustainable. But what we thought is like, let's try to form these pores in a planar lipid bilayer setup. So with, let's say, 1 year of struggle with different ratios and all these things, we found finally a perfect recipe to form these pores. So with a surfactant, we call it Ampifold. It gives amazing stability to the pores, and especially if it is large, you need something which gives stability of your pore for enough time, which gives you enough window to measure all the samples for in a reliable manner. So we found the ratio and all these things. And if you look at the current recordings at the nice stoichiometric ratio, you see with time, this is like a stable baseline. And then once, like within 10 or let's say 5 minutes, or sometimes within seconds, it forms this huge current jump, which is like a single step insertion. And then if you measure the diameter, it's like 20 nanometers. This is like an amazing pore, and it's stable for 30 minutes. And if the bilayer is stable, sometimes it's stable for 1 hour or so. So it's like, we were quite shocked when we first saw this pore and the stability of the pore. And we have also done the IV curves, which gives us like it behaves perfectly like a cylindrical pore, which we have a perfect ohmic nature of the IV curve. So since we have the pore, we thought, okay, first we have to calibrate the pore if it can do a good job in analyzing known proteins. For example, folded proteins with known molecular weight and known size and shapes. So we added different molecular weight proteins from top, and you see, we generally see this kind of spikes. And then if you enlarge the spikes, it looks like this. And from the current modulation, basically you can see, so we have chosen four different or five different proteins. And we see from here, the dotted lines are the expected volume or the sizes from its PDB structure. And these are the measured ones from our nanopore measurements. So you see that's nicely correlating with the model one. And here is just an example. Our volumes and then the reference volume, it's almost like perfect matching. Like if you get a nice straight line for both the size and also aspect ratio, or we call it length to diameter ratio. So all this is good. Now we have a nice pore which is calibrated and all. So now we can move on to our original goal of analyzing tau protein. So if we have chosen for this purpose the longest tau available for Alzheimer's is called tau 441. It's a full length tau. And so under different laboratory conditions, for example, we have aggregated the protein, and then within 1 hour of aggregation and 2 hours of aggregation, you can see that in the TEM images, they form this nice clumps of proteins, which are spherical, like which are oligomers, but not exactly fibrils in the structure. And we thought, and you can see also from the images that these are of all different sizes. So this is a perfect example of an heterogeneous samples and to test our pore. And here exactly how our measurement look like. So you see here from tau 0 minutes to 1 hour to 2 hour, we saw the amplitudes. And then if you analyze it nicely, you see that with 0 time to 1 hour to 2 hour to 3 hour, you see this is O1 is like the monomer. So the monomer population gradually decreases. And then you see an increase of dimer and eventually trimer, tetramer, and pentamer, and all these oligomers. So this is by today, I'd say the first example where you can see all these different populations nicely for the first time in a single molecule measurements with a lot of good number of statistical data points. Because the larger oligomers are quite, you can see it from TM and all other techniques, but these are for the first time we are showing for the very early oligomers of tau. And the advantage of our technique is not only we can find the molecular weight or diameter of the oligomers, but also we can analyze the length to diameter ratio, and you can see from here to monomer to dimer to trimer, it is going to a more spherical structure. So from this is very important because this can be one of the first example where we can correlate how exactly the tau oligomer forms from monomer to before the fibrils, and then it forms. So it's like the starting point of the fibrils and then how it exactly grows. This is basically it. So to summarize, we have a very nice pore, 20 nanometer, and then we are also working on different new pores, which I cannot talk about right now, but we have many large, like quite a bit of large pore than this. Now at this point, just like the individual, like the very preliminary experiments, and also for all the tau or neurodegenerative experiments, we also have done, so this is for tau oligomers in situ assembled in our lab, but we have also tried experiments with tau samples of the Alzheimer patient serum samples and blood samples of Alzheimer's and also Parkinson's. I don't have the data right now because these are all preliminary data, but we can. So since we have all these results now, we are trying to correlate with the real sample of the serum sample and the blood samples, and we're trying to see exactly how these populations are. And then we're trying, since we know the shape or the aspect ratio of these populations, we're trying to cherry pick all the ones from the early ones and trying to correlate them both volume or molecular weight wise and also shape wise. It's very early work. Maybe in the next nano meeting I can show this. But yeah, these and things, and also thanks to the group in Adolf Mackley Institute, University of Freiburg, and also thanks I'm very much thankful to NCCR for funding our research, and also Novartis and also Swiss National Science Foundation for funding most of our Alzheimer's and Parkinson's work. And thanks to you for the time, and I'll be happy to take any questions.
Thank you so much, Anasua. Are there any questions from the audience?
Hi, thank you very much for this very interesting talk.
Thank you.
You mentioned that your pore is stable for half an hour up to an hour. What is actually happening after that? So is it getting noisy or closing, gating?
Yes.
What does it do?
Yeah, very interesting question. So what we believe is that after half an hour, 1 hour, our bilayers are not stable, because we generally see the bilayers are breaking. Like, for example, it goes to an offset, because we have like optimized the condition so that we know that there is only one nanopore which can form, and we generally avoid forming multiple pores. So we know that maybe this bilayer is not breaking. We don't know exactly, but we are suspecting that the bilayers are not stable after an hour at this applied potentials.
Thank you. It's very nice talk. I'm wondering what's Amphipol doing with bilayers. Have you done controls or?
Yes. Yes, thank you so much for this question. This is actually, so if you look at the cryo-EM structure, okay, I don't have the things here, but exactly what Amphipol does is Amphipol acts like an anchor, and it stabilizes. So there are cryo-EM evidences that whenever they're measuring the diameter of the pores, it's very uniform if they have Amphipol in the lysosomes. So what we believe is that they act like, for example, these red ones, they act as anchor and holds the hydrophobic regions of the protein, like beta barrel structures in the cylindrical form here. And I think so they are just stabilizing this the membrane with the pore, since these are large pores. It just enhances the stability.
Like after an hour, because in our experience, we see or we can measure 4 hours. Well, if you can
really?
just self-assemble pores like Pneumolysin or pyrolysin, so we don't see any break like in half an hour. So you can measure like 5 hours or more.
Yeah.
So that's why I'm wondering if it's Amphipol what's making membrane unstable, because it's associated.
Okay, because we also have control experiments where before, like if we don't add any pore formers, then the bilayers are stable. So probably, I don't know, maybe the bilayers are not breaking, or probably the pore is just out, or maybe it's just breaking the bilayers.
Synthetic membranes like polymer membranes or
Yes.
membranes.
Yes. We tried actually polymer membranes. Now we are trying to move away with amphifols, because amphifol also creates sometimes some issues with other proteins, because if we have analytic proteins and then amphifol is a surfactant and it can also bind. So we are trying to move away from amphifols. But what we have noticed is for our other pore, which we call like a polysinine pore, works wonderfully well in synthetic membranes, but not this Pneumolysin pore. Probably these are, I don't know, we don't know exactly the reasons for this, but we don't know. It will be interesting to know, but we don't have any reason for this right now. But thank you for the inputs.
Many thanks for your talk. Maybe I lost something, so I'm sorry for my question, but how much is selective your pore against other proteins?
Yes. So right now, the pores are not selective. So right now, if you have like a perfect molecular weight size which passes through the pore, all proteins will pass and you will characterize everything. So what we are right now working on is that we are trying to attach some nanobodies on the top of the pores, so that and those will be like selective for all these biomarkers, and then we are trying to enhance the selectivity in this way. But it's difficult when you have a mixture of proteins and nanobodies and assembling things. Probably in a year will be there, but that's the ultimate goal, to make it selective for some really interesting proteins.
Okay, excellent. So I just wanted to know, is the success rate actually horrific, or is it easy to get such a
This
pulse?
is really good. Like, for example, if you
20
use
nanometer.
yes, so if you, for example, use orbit 16 within
Not what I wanted to get.
use so within I think so 10 minutes or 20 minutes, you'll get at least 5 or 6 different pores.
Excellent.
And
Right.
this is what it is.
Okay, yeah, maybe one final question.
Yes.
Do you always get the same size of current? I mean, the same conductance? I mean, I would assume that there's a lot of
Yes.
variation because you have so many
Yes.
monomers.
That's true. That's very interesting. Yes, generally speaking, we do not get the same diameters. So, for example, for this pore, we have a standard deviation of 3 nanometers. So the pore diameter varies from 20, like plus minus 3. But then we have developed a code where each time for live monitoring single UCI insertion, we can calculate the diameters.
Okay.
And then we calculate at the same time the diameter of the pore, and then we choose that we give input these values for the analysis.
Okay, so your analysis can adapt to different types
Exactly.
of pore.
Yes.
Okay, yeah.
We have changed the analysis in this way.
Finally, this graph you showed on number 19,
Yes.
you plot on the x-axis, you plot molecular weight.
Exactly.
So how do you arrive at the molecular weight from your signal? Because this is derived from, I mean, this must be a rather complicated
Exactly. This is
derivation, no?
yes, it's very interesting. Yes, exactly. Right now, this is what we are discussing, and also in the paper's method section.
Okay, but maybe you have to.
Yes, but then I can tell you simply what we are doing is like we from our nanopores, we can get all the volume values, okay? And then we know the shape. For tau, it's really interesting, and you can see also in the TM images that they're spherical. So we are assuming it as a spherical particle, and then we are converting the spherical volume to a molecular weight with the known published formula. So it's like R is equal to 0.066 cube root of m molecular weight, and then we are getting all the numbers. But yeah, it will be interesting if it's a very anisotropic protein, then we have to just assume it as a spherical protein for the molecular weights. Thank you.